I took him for a tour of one of the most exciting parts of the brain when it comes to neurogenesis. And this is the hippocampus. So this is this gray structure in the center of the brain. And what we know since already very long is that this is important for learning and memory, and mood and emotion. However, what we have learned more recently is that this is one of the unique structures of the adult brain where new neurons can be generated. And if we slice through the hippocampus and zoom in, what you actually see here in blue is a newborn neuron in an adult mouse brain. So when it comes to human brain, my colleague Jonas Friesen from the Karolinska Institute has estimated that we produce 700 new neurons per day in the hippocampus. So you might think this is not much according to the billions of neurons we have, but by the time we will turn 50, we will have all exchanged the neurons we were born with in that structure with adult-born neurons. So why are these new neurons important and what are their functions? So first, we know that they're important for learning and memory. And in the lab, we have shown that if we block the ability of the adult brain to produce new neurons in the hippocampus, then we block certain memory abilities. And this is especially new and true for spatial recognition. So like how you navigate your way in the city. So we're still learning a lot, and they are not only important for memory capacity, but also for the quality of the memory. And they will have been helpful to add time about memory, and they will help differentiate very similar memory, like how do you find your bike that you park at the station every day in the same area, but in a slightly different position. And more interesting to my colleague Robert is the research we have been doing on neurogenesis and depression. So in animal model of depression, we have seen that they have a lower level of neurogenesis. And if we give antidepressants, then we increase the production of these newborn neurons, and we decrease the symptom of depression, establishing a clear link between neurogenesis and depression. But moreover, if you just block neurogenesis, then you block the efficacy of the antidepressant. So by then, Robert had understood that very likely his patients were suffering of depression even after being cured of their cancer, because the cancer drug had stopped the newborn neuron to be generated. And it will take time to generate new neurons that reach a normal function. So, collectively, now we think we have enough evidence to say that neurogenesis is a target of choice if we want to improve memory formation or mood, or even prevent their decline associated with aging or associated with stress. So the next question is, can we control neurogenesis? The answer is yes. And we are going to do now a little quiz. So I'm going to give you a set of behavior and activity, and you tell me if you think they will increase neurogenesis or if they will decrease neurogenesis. Are we ready? Okay, let's go. So what about learning? Increasing? Yes. So learning will increase the production of these new neurons. How about stress? Yes. Stress will decrease the production of new neurons in the hippocampus. How about sleep deprivation? Indeed, it will decrease neurogenesis. How about sex? Oh, wow. Yes, you're right. It will increase the production of neurons. However, it's all about balance here. We don't want to fall in a situation about too much sex leading to sleep deprivations. So how about getting older? So neurogenesis rate will decrease as we get older, but this is still occurring. And then finally, how about running? So I will let you judge that one by yourself. So this is one of the first studies that was carried out by one of my mentors, Rusty Gage from the Salk Institute, showing that the environment can have an impact on the production of new neurons. And here you see a section of the hippocampus of a mouse that had no running wheel in its cage. And the little black dot you see are actually newborn neurons to be. And now you see a section of the hippocampus of a mouse that had a running wheel in its cage. So you see the massive increase of the black dots representing the new neurons to be. So activity impact on neurogenesis, but that's not all. What you eat will have an effect on the production of new neuron in the hippocampus. So here we have a sample of diet, or nutrients that have been shown to have efficacy. And I'm just going to point a few out to you. So calorie restriction of 20 to 30% will increase neurogenesis. Intermittent fasting, so spacing the time between your meal, will increase neurogenesis. Intake of flavonoid, which is contained in dark chocolate or blueberry, will increase neurogenesis. Omega-3 fatty acid present in fatty fish like salmon will increase the production of these new neurons. Conversely, a diet rich in high saturated fat will have a negative impact on neurogenesis. Ethanol, intake of alcohol, will decrease neurogenesis. However, not everything is lost. Resveratrol, which is contained in red wine, has been shown to promote the survival of these new neurons. So next time you're at a dinner party, you might want to reach for this possibly neurogenesis neutral drink. And then finally, let me point out the last one, a quirky one. So Japanese groups are fascinated about food textures, and they have shown that actually soft diet impairs neurogenesis as opposed to food that require mastication, chewing, or crunchy food. So all of this data, where we need to look at the cellular level, have been generated using animal model. But these diets have also been given to human participants. And what we could see is that the diet modulates memory and mood in the same direction as it modulates neurogenesis, such as calorie restriction will improve memory capacity, whereas high-fat diet will exacerbate symptoms of depression, as opposed to omega-3 fatty acid, which increase neurogenesis and also help to decrease the symptom of depression. So we think that the effect of diet on mental health, on memory and mood is actually mediated by the production of the new neuron in the hippocampus. And it's not only what you eat, but it's also the texture of the food when you eat it and how much of it you eat. On our side, neuroscientists interested in neurogenesis, we need to understand better the function of these new neurons and how we can control their survival and their production. We also need to find a way how to protect the neurogenesis of Robert's patients. And on your side, I leave you in charge with your neurogenesis. Thank you.
Okay, now you know this. This is actually very important. People are very have strong opinions about antidepressants. In our culture, usually people older than 50 are strongly against antidepressants. They say that they make you addicted. You become addicted to antidepressants, and so it is best not to start it. First of all, antidepressants are not addictive drugs. Addiction is something else. And second of all, depression is a huge epidemic right now. You may know, you may realize, you may be experiencing depression as well. Depression is not about your condition or is not about what you are thinking, but it seems like that. Like when you're in a depression, you think that you won't get out of that state because of the state that you're in. Like, For example, you can say that my life is like this, my situation is like this, so this is the result. As a result, I'm in depression. It's not like that. Depression is actually a very how to say it? It has a very simple mechanism. Your brain actually stops producing new cells or decreases producing new cells, and it puts you into depression. And also vice versa, when you are in depression, your cells stop proliferating that much. And when you take antidepressants, it immediately accelerates the production of new cells in your brain. And this is well known about Prozac. Prozac is a selective serotonin reuptake inhibitor, and there are other versions of that drug, SSRIs. There are many, for example, Lustral, sertraline, or Cipralex, for example, most commonly used. Prozac, as you already know, is fluoxetine, the etken madde, main thing in it. So, if you have older people around you who is suffering from depression, you may choose to start on antidepressants, obviously after a doctor's, a physician's consent. They have to write it down. Actually, antidepressants, because they are increasing the neurogenesis in your brain, it has an anti-aging and has a protective role in Alzheimer's or Parkinson's and stuff like that. And if you induce sleep, like sleep drugs in elderly people, because it induces sleep, and sleep is a hugely reparative, like it repairs the brain. So if you induce sleep, it's a good thing as well. So you can increase your awareness about this drug. So I'm so much pro antidepressant person. I'm using antidepressants myself. It's a disclaimer. I'm not like urging you to start antidepressants, but it's not something to be feared of. If under a doctor's control, like under prescription, you can use them safely, and it doesn't do you any good to stay in the depressive mode. Like depression is something. One of the worst things that can happen to your brain. It slows down your brain's functions, it slows down your brain's production of new cells. It has actually, depression has an evolutionary role, because when winter comes, body or brain enters into a depressive mode so that you won't go outside and you won't risk your life as an evolutionary structure, because in the winter you can be eaten by wolves outside, but now you won't be eaten by wolves, and you have to continue your life even if there is too little sunlight. So you can get some help, and therapy is also a good option. Therapy, how it works is when you take the antidepressants, you produce new cells, and if you are going to a therapy, because they are teaching you a new ways of thinking, so your new cells adopt the new ways of thinking, so it helps you as well. So antidepressants plus therapy plus social support systems. Social isolation is also something bad for your brain. Do not stay at home isolated from your environments. Go outside, interact with people, take some sunshine, and take yourself out of depression. Depression is not something good. I won't suggest you to stay in depression so very long. It's not healthy. And if you have elderly people around you, you can inform them as well. Okay. So, glycoprotein as a carbohydrate group covalently attached to a protein. We have lots of glycoproteins. Where they are located? They are located in the cellular membrane, glycoproteins. And they make up the code of the cell. So the cell can be recognized from outside by looking at the carbohydrate groups. The immune system also recognizes the cell by looking at glycoproteins. And glycosylation increases the complexity of the proteome. What does it mean? You have lots of proteins. How many proteins do we have? Around 20,000, I think. 20,000 proteins, but glycosylation also increases their variation, different glycosylated forms. Proteoglycan layer is a carbohydrates plus proteins. It has as a function as structural component, like it is important in bacteria mostly. And also mucins, which I was producing last week. It's the mucus. It is the lining of your internal organs like the lungs and bronchia and all, and your it is in Turkish sümük and mucus. It's mainly actually sugars. Korkunç bir şey söyleyecektim ama vazgeçtim. So it has sugar moieties in your mucous system. It has the innate immune system, you may remember. The mucus that you are producing and running through your mouth actually prevents bacteria from entering. And whenever you have a disease that prevents the production of mucus, it dries inside, like your airways. If it dries up, then you are more exposed to the bacteria coming from outside or viruses. So it is the innate immune system. And linkage or linkage, again we are coming to that. And linked carbohydrates can be added to the proteins through nitrogen linkage or oxygen linkage. So oligosaccharides can have some glycoproteins can have a structure like this. For example, this is mannose here, and then this is an acetylglucosamine, this one, and glucose, stearic acid, galactose. So here we have the protein from here, and then here we have the carbohydrate added to the proteins. And linkage is in here, where the carbohydrate attaches itself to the protein. We have the linkage there. Erythropoietin is a glycoprotein. It's secreted by the kidneys into the blood, serum, and it stimulates the red blood cells. It is abused by some of the endurance athletes because if you take erythropoietin, it immediately starts producing red blood cells, and so it's a doping. It's a doping material. Glycosylation enhances the stability of the protein in this case. Now this is erythropoietin structure. This is the protein part, and then lots of carbohydrates are added. Almost 40% by weight is carbohydrates. Now, in nutrient sensing is a mostly more critical, like it's critical for single cell organisms because they have to sense where there's a nutrient and then move towards the nutrient. In our case, it's not that critical, but still it is embedded in our cells, and there are like nutrient sensing pathways, which is called mTOR, mammalian target of rapamycin. It's called mTOR pathway. It's a nutrient sensing pathway, but we have these acetylglucosamine. This one, I can't pronounce this. It's for sensing nutrients in the membrane. They are also phosphorylated, and regulation of these things, I forgot it, acetylglucosamines, they are linked to insulin resistance, diabetes, cancer, neurological pathologies. This is a kit. Like whenever there is a critical thing damaged in the cell, we immediately say that it may relate to insulin resistance, diabetes, cancer. It is actually comprises all the diseases, but actually these are called metabolic syndromes. This is metabolic, and these are the results. These are affected by the metabolic syndromes as well. it means that in your carbohydrate metabolism, mostly, there is something wrong which leads to metabolic diseases. Proteoglycans we already saw. So glycosaminoglycans, these are, you can find them in cosmetic industry. Hyaluronate is present in almost all facial creams right now. Hyaluronate. This is very common. If you go to Watson's, and then you can see on each of the facial creams hyaluronic acid is added. But also it's a very good eye drop. Sunni gözyaşı, they're small tubes that you just use once and drop in your eyes. It is also hyaluronate, by the way. It lubricates your eyes. Dermatan sulfate is also present in some of the creams. Heparin is kan sulandırıcı, blood thinner. These are all have some applications, and these are all in the family of glycosaminoglycans. Cartilage, kıkırdak, has collagen. Collagen is also huge in cosmetics industry, you may know. There is a ongoing debate. If you put collagen as a cream onto your body, does it get into your cartilage? This is the question.
No.
Nobody knows this. Yeah, it's probably no. If you take collagen as a supplement, does it go and replace your cartilage? Probably again no. But what is very rich in collagen?
Bone broth.
Yeah, kelle paça, kemik suyu. It is very rich in collagen proteins, and it is highly recommended during illness, and also as an anti-aging strategy, but I'm not sure if it helps. But what is important is that kıkırdak, the cartilage, you lose your cartilage tissue while you are getting older, and if you don't have the cartilage tissue, the bones just has a friction in between them, and because your bones are actually like breakable things, they tend to break easily if you don't have enough cartilage. So collagen is the main thing in your cartilage structure. This is cartilage, how it is composed of. It has hyaluronate, keratin sulfate, and then this is called aggrecan because it has like, it's an aggregate type of thing, and it holds up water so that the water structure can make it more flexible. The more flexible the structure, the less it will break, so it absorbs the pressure. Osteoarthritis is an aging disease. It means that water is lost from the proteoglycan layers, so you have this kireçlenme as a disease. Mucins, we already saw, these are the mucus structure, keeps your lungs hydrated. So overexpression occurs in bronchitis, cystic fibrosis, and adenocarcinomas. Do you remember cystic fibrosis? They produce a lot of mucins in their lungs, so they have this constant
Coughing
and
discharge.
mucus producing. Yes, exactly. If you don't have the mucus, it prevents breathing. If you have too much mucus, it also prevents breathing. So for the breathing to be continued nicely as a healthy way, there is a level of mucus production. And then protein glycosylation, we need endoplasmic reticulum and Golgi. N-linked Glycosylation happens in ER and then continues in Golgi. Only glycosylation occurs only in Golgi. So Golgi and endoplasmic reticulum has some duties separated in between them. Blood groups are protein glycosylation actually. There are antigens in blood groups, different carbohydrates. They are actually different carbohydrates. A and B antigens, there are A and B antigens. Type A transferase acts an acetylgalactosamine to form the A antigen, and type B transferase acts galactose to form the B antigen. So all that matters between the different blood types is only one carbohydrate is in here. So this is the O antigen, this is the base antigen. So in A antigen you have this, and B antigen you have this galactose or galactosamine. So a person who has A antigen produces A antigen. So zero blood type actually lacks both enzymes. AB has the both enzymes. A has A transferase, B has B transferase. So if in the blood transfusion an antigen enters your system, your blood recognizes it as foreign and then it produces a huge response. This is actually not this simple. I saw a blood bank, Kan Bankası Kızılay. They have a panel of tests that they do on characterizing the blood, and then they separate the blood thread part and the serum part, and then they look, I think like 40 different types of tests that they do on bloods, and it has to match. So you cannot say like, I'm with the same blood type, just transfer my blood. It only happens in movies, I think. So bloods actually come from the blood bank after a series of testing. Obviously they test for viruses like HIV, HCV, HPV type of, but they also have other factors that they look for apart from these types. Cholera. Apparently people with blood type 0 are 8 times more likely to have the severe disease because if the antigen binds to the toxin other than the other blood type antigens. Congenital disorder. Congenital means that it's coming from birth. They are pathologic conditions. There are some muscular dystrophies, like muscular dystrophy means that the muscles lose their tension over time. There are lots of muscular dystrophy diseases. Some of them are linked to glycosylation. For example, eye-cell disease is a lysosomal storage disease. It has something wrong in glycosylation. To separate oligosaccharides or to identify them, Malditov is used as a technique. Malditov is an HPLC chromatography technique. It can differentiate between different oligosaccharides because mass spec is already a very sensitive protocol. So it can separate between the carbohydrates as well. Lectins, glycan, if a protein binds to a carbohydrate residue, if it can bind to it, then it is called lectin. They facilitate cell-to-cell contact because they bind to the cell displays carbohydrate on the membrane. So if a protein can bind to it, it can recognize that cell. So lectins are important in cell recognition. Influenza virus has a lectin protein that binds to carbohydrates on the cell surfaces. So it binds to the carbohydrate on the cell membrane, and then it attaches itself, and then when it is engulfed into the cell, it enters the host cell. So the virus can enter that way. Like this. So, vallahi erken bitirdik koca chapter'ı. Because the whole chapter is a series of keywords and definitions. There is no mechanism. And I couldn't find any animation regarding that. There is not a nice animation because it's all definitions. So I will try to find some creative questions and upload them to Moodle. On Friday we can start with it. Any questions? Final kümülatif olacak ama ağırlık son konulardan olur. Bu genel soruya da cevap vereyim. Okay then.
Ben gitmek istemiyorum. Ona gideceğim. Efendim bir şey yap. Oturup oynayalım. Yazmam ders. Gel.